Co-Diagnostics has developed a solid strategy for dealing with the COVID-19 pandemic over the next few years, which includes engineering products that will enhance the ability of the world's schools, businesses, and hospitality industries to address the stark reality that COVID-19 illness is likely here to stay. We're not going to be vaccinating our way out of this to 8 billion plus people in the world right now, and ease of transmission, along with new strains, means that COVID-19 testing needs to become part of the long-term plan at many organizations in our society and the world at large. Helping to provide affordable, accurate, and fast diagnostic solutions for COVID-19, flu, and other diseases is what Co-Diagnostics is all about. In addition, we're excited about our initiatives in liquid biopsy for cancer detection and the important genomic work we're doing in the ag biomarket. In this presentation, we are pleased to update you on some new technological advances in direct saliva protocols for COVID-19. And I believe you'll find what our scientists have to say to be important and very interesting. Co-Diagnostics has successfully demonstrated that the Co-Primer platform technology can be used to identify the presence of SARS-CoV-2 in human saliva samples without first requiring RNA extraction of the sample, and can do so while providing low limits of detection. This technology is a significant advance, and eliminating the costly and time-consuming RNA extraction step allows for even greater speed and economic advantages. We believe this discovery speaks to the strength, innovation, and flexibility of our Co-Primer platform, not just as it relates to creating improved testing for technology for COVID-19, but also additional possibilities to implement Co-Primers in various other diagnostic applications from point of care to high-throughput centralized lab settings. Our next steps include incorporating extraction-free direct saliva tests into the company product offerings, including those for COVID-19, so they can be available to our customer base worldwide. With that, I'll turn the time over to our scientific team.
Hi, I'm Dr. Casey Bramwell, Director of Research and Development at Co-Diagnostics, and I'm happy to take this opportunity to discuss some of our work on direct saliva testing with Co-Primers. To put this work in context, as of February 2021, the U.S. has conducted well over 100 million PCR tests for COVID. Traditionally, each of these tests requires an extraction prior to the PCR reaction. This extraction step adds extra expense, requires independent reagent supply chain and instrumentation, it increases the time to result, and reduces the overall testing throughput. For all of these reasons, the U.S. Food and Drug Administration has identified the need to reduce reliance on testing supplies as an ongoing priority. COVID testing is compatible with several different sample types, but saliva has the advantage that it is relatively easy to collect, it can be self-collected, and it is already utilized by numerous tests with FDA emergency use authorization. Several groups published direct sample testing protocols with saliva, prompting us to evaluate the performance of Co-Primers with an extraction-free, minimal processing procedure. As depicted, this processing is relatively simple, with sample collection, addition of proteinase K, vortexing to mix, heating the sample, combining the sample with PCR reagents, and then running the RT-PCR reaction to test. As we implemented the minimal processing protocol, we evaluated the impact of modifying the order of operations. We found that proteinase K performed several important functions: to release viral nucleic acids and to inactivate endogenous protective enzymes that are present in saliva, and that this proteinase K treatment must be followed by heat inactivation prior to PCR testing. We know that our chemistry is compatible with many different instrument platforms and assay configurations, so we evaluated the performance of Co-Primers with several different reaction volumes and master mix concentrations. With 6 different conditions, we had very consistent performance. Using 15.6 copies per reaction of synthetic material, every condition detected 20 out of 20 replicates. So we tested at a lower concentration, where we again found consistent performance with all conditions, detecting 18 or 19 out of 20 replicates. When we calculate the associated limit of detection, we found analytical sensitivity to be as low as approximately 350 copies per mil. in the 30 microliter reaction using a 4X master mix. Our results for this and other studies that we completed during feasibility testing are available in our white paper published in November of last year. Since then, we've been hard at work developing a new product, the Logix Smart SARS-CoV-2 DS test. This is a real-time multiplex RT-PCR test using our proprietary coprimer technology with minimally processed saliva. It targets two highly conserved genes within SARS-CoV-2, the RDRP and E genes. Each kit includes a ready-to-use 2X master mix using RNase-P as an internal sample quality control, a positive control to verify the performance of the test, and nuclease-free water as a negative control to verify lack of contamination during testing. We have validated the test across numerous platforms using a 20 microliter reaction volume. The validated instruments include the CoDx box, the MIC from Biomolecular Systems, the ABI QuantStudio 5 and 7500 FastDX from Thermo Fisher, and the CFX96 from Biorad. Although the test has been validated by Co-Diagnostics, the US FDA's independent review of this validation is still pending. Here we have depicted our analytical sensitivity using numerous different platforms, which each compare favorably with that of other published direct saliva assays, which typically range between 6 and 12 copies per microliter. We achieved very high clinical sensitivity, with 97% concordance among positive saliva specimens that went through extraction prior to testing, and 100% concordance among negative samples. Our in silico inclusivity is also extremely high for both target genes, which we will hear more about momentarily. Next, you'll hear from our lead laboratory scientist, Madison Stark, who will take you through the direct saliva testing process.
I'm Madison Stark. I'm Co-Diagnostics lead laboratory scientist, and I'm going to demonstrate the ease that the Co-Diagnostics Logix Smart Direct Saliva can be implemented into any laboratory size. The protocol begins by vortexing your saliva sample to help with any viscosity. You will then add 2.5 microliters of proteinase K into a microcentrifuge tube, strip tube, or plate, depending on what laboratory you're operating at, along with 50 microliters of your vortexed saliva. You will then cover your samples, briefly centrifuge, and then vortex for 1 minute. After vortexing, you will briefly centrifuge your samples again, and then place them in the heat block or thermocycler to sit at 95 degrees for 5 minutes. Allow your sample to reach room temperature before plating with your Co-Diagnostics Logix Smart reagents. The samples can then be processed through normal PCR, just like any other extracted material. That's the simplicity and beauty of using the Co-Diagnostics Logix Smart Direct Saliva.
Hello. My name is Mason Christensen, and I'm the Senior Design Engineer for Co-Diagnostics. I designed the markers that we're using on our COVID-19 tests and the direct saliva test that we're discussing today. I also perform ongoing in silico inclusivity and exclusivity analyses on these markers, and will be summarizing the results of those analyses during this presentation. Inclusivity, meaning that we include everything that we want to, in this case, all strains of SARS-CoV-2, including ongoing mutations and emerging strains, and exclusivity, meaning that we exclude everything else. For exclusivity analysis, we used the BLAST-N tool to query the entire NCBI and associated databases. We have 2 markers for our COVID tests. For the RDRP marker, there was no significant homology with any other organisms, except for some strains of SARS found in bats, which had as low as 1 mismatch on the binding target. We don't expect decrease in sensitivity due to a single mismatch, so if you are testing bats with SARS, then that would lead to a false positive result. We do not consider this a clinical risk, however, since our tests are intended for human use. For our E gene marker, the results were similar for bats with up to complete homology, and also complete homology with some samples of SARS-CoV-1. Again, we don't consider this a clinical risk, since there have been no cases of SARS-1 infection since that epidemic ended over 15 years ago in 2004. Since no other organisms in the database came up as significant matches, we do not expect any cross-reactivity that would affect our clinical specificity or result in false positive tests. We also specifically analyzed homology with each of a series of high-risk relevant organisms, including other coronaviruses, high-priority organisms that may be present in our sample collection, and other organisms of importance, such as influenza and others. Again, we found no homology or prediction of cross-reactivity, with the exception of SARS-1, as discussed previously. For in silico inclusivity, we are employing 3 main systems of analysis to make sure that we include all strains of COVID-19. I will summarize here, and then we will go through each in more detail. First, we use the Next Strain tool, which pulls from the GISAID database to perform monthly analyses of the global subsampling that pulls a statistical sample from sequences all over the world. We also perform our own analysis, pulling strains from the NCBI database, most recently analyzing nearly 41,000 strains and their relation to our markers. Finally, we individually analyze emerging strains as they are identified as potentially problematic, recently looking at the new variants coming out of the UK, South Africa, and Brazil. We do not predict any loss of sensitivity or specificity of our markers relative to these strains. For our monthly analyses, using the Next Strain subsampling, this is an extremely useful tool showing predicted phylogenies here, countries of origin, including nearly every country worldwide here, and at the bottom, they compile location and frequency data for the mutations on each of these strains. Using this, we are able to look at the binding targets of our coprimers and analyze mutation data relative to our tests. So, for the results of those analyses. For our RDRP marker, we have those monthly analyses from the beginning of the pandemic, where we had 100% homology with all sequenced strains, and as the months progress and mutations start to arise, we have them here. Until now, there are about 1% of strains with a single mutation on our target region. As I mentioned earlier, we don't expect to see reduced sensitivity due to a single mutation. There will be just a slight delay in the PCR cycling, but 2 and especially 3 or more mutations is where we expect to start seeing sensitivity decreases. And those are so rare that we actually have not seen any in these analyses for our RDRP marker. For the E-gene marker, the results are similar. There have been a few times where we observed 2 or more mismatches, but at a rate that is low enough to be considered statistically insignificant. For this marker, there is a lower rate of single mutations, currently sitting at about 0.5%. Since we do not select the sequences for the Next Strain global analyses, we wanted to perform our own analysis to see if it would corroborate the results. We downloaded about 41,000 complete SARS-CoV-2 genomes from the NCBI database and compared them with our markers. We repeated this analysis yesterday, February 8th, so those are the results that you'll be seeing now that are up to date. We aligned the sequences using viral MSA, which uses Minimap2 as its aligner. For the results, for the RDRP marker, this shows cumulative mismatch percentage on the y-axis over the sequence analyzed. The x-axis shows genome location with our forward coprimer binding target in red and the reverse in blue. I won't be going over coprimer structure during this presentation today, but they do have 2 binding targets each. Their function is similar to traditional primers, with the exceptions that their structure results in increased specificity, virtually eliminates primer dimers, and eliminates the necessity for a fluorescent probe, since we are able to label the coprimer itself while still facilitating amplification. For these mutations, we would start to consider them significant if they began approaching 1%, or especially 3% or higher. As you can see, none of the mutations in this region are close to that threshold. The E-gene marker has a very similar results, again, with the most common mutation occurring about 0.2% of the time. So the results from our own analysis do support the conclusions from the Next Strain analysis, that mutation on our tests are rare and not significant enough to cause predictions of reduced test sensitivity. Let's also discuss in more detail the emerging strains being identified out of the UK, South Africa, and Brazil, 501YV1, V2, and V3. These strains may possess higher transmissivity or ability to spread. They are primarily characterized by mutations on the S gene, which codes for the spike protein. Since our markers target the RDRP and E genes, those mutations are obviously irrelevant to our test. We also analyzed each strain individually 4 mutations across its full genome, and none of them are on or near the binding targets of either of our markers. Therefore, we do not predict any effect on our test due to these variants. Also, in these next strain global analyses, you can see that those 501Y, V1, V2, and V3 variants are also highlighted here, so they are included in our ongoing surveillance as well. In conclusion, we expect near zero cross-reactivity or false positive results due to other organisms, infectious diseases, or other DNA or RNA in test samples. We also expect near zero false negative results due to mutations and genetic drift of the SARS-CoV-2 genome, including on emerging variants. Since this has been stable for the past year, and since all indicators are well within acceptable ranges, we do not expect this to change in the near future. However, we will continue performing surveillance and monitoring the situation, and will be ready to respond quickly in the event that something does change in the analyses. Thank you.
As a company, we agree with Dr. Michael Osterholm that we will be dealing with COVID-19 till the end of time. From here on out, we will be dealing with the flu, cold, and COVID-19 season for decades to come. With this inevitability ahead of us, we have engineered and are currently developing robust products that we believe will play an important, ongoing role in dealing with these upper respiratory diseases. The company has developed a robust COVID-19 test menu, including a new multiple gene assay targeting international customers, and our powerful ABC assay that detects flu A, flu B, and COVID-19 in one easy-to-use and easy-to-interpret test that is highly accurate. Along with our direct saliva technology demonstrated in this presentation, we look forward to providing robust solutions that are affordable, fast, accurate, and accessible worldwide. Thank you for taking the time to learn more about Co-Diagnostics today, and please visit us at codiagnostics.com or email us at info@codiagnostics.com.